Hello, this is Miss Cortez, and I am your biology teacher. So, what we are going to go over is protein synthesis and base mutations. So, as you guys have already talked about the central dogma of biology, replication, transcription, translation, protein synthesis is mainly going to focus on those last two steps, transcription and translations. With base mutations, these are going to be mutations that happens within these two. God bless you, Joaquin, these two last steps that could cause different mutations, which cause different outcomes in proteins. So we'll talk about it. Okay, so protein synthesis. What does the word synthesis mean? So synthesis is just basically a fancy word for make. So if we're talking about this process, we're just talking about protein making. So this is a process by which the cell creates proteins through two processes, transcription and translation. So if you look at the GIF down below, right now what we're seeing is translation, where tRNA comes in on the mRNA, meets up, and is able to drop amino acids. Amino acids all connected together form a protein strand. Okay, now we're looking at transcription here, where RNA polymerase comes along the mRNA and makes a copy of a gene. Genes code for different proteins, and that segment of DNA it's copied into mRNA is what is able to make the protein. So key terms. So mRNA, if you remember, is messenger RNA. tRNA is transfer RNA. Our RNA is ribosomal RNA. Proteins are structural components of living organisms. These are our building blocks that we talked about when we talked about macromolecules. So this is what our muscles are made of. This is what our enzymes are made of. So these are incredibly important, our proteins. So this is why we care so much about protein synthesis. This is what makes us up. Okay. From there, our amino acids, these are the building blocks, also known as monomers, so our smallest parts of our proteins. A codon is what is going to be on our mRNA. So it is 3 consecutive nucleotides, so 3 bases on the mRNA that code for a specific amino acid to be brought in on the tRNA. Our anticodon is just that matching code, that complementary code on the tRNA that's bringing in the amino acid. So transcription. Transcription is where the mRNA copies a message from the DNA to make a protein. So this message is usually specifically a gene that is coding to make a specific protein. Okay? So this occurs in the nucleus of a cell in a process that is similar to DNA replication. So our RNA polymerase is going to go through, our DNA is going to be opened, and we are going to make a new strand of mRNA, RNA off of our DNA. And the thing to remember about this process is in mRNA or any of our RNAs, we are not using thymine, we are using uracil as our base instead. With translation, okay, this is our next step. So after our mRNA is made, it is able to leave the nucleus, which our DNA cannot do. So it goes out into the cytoplasm, which is just the inner fluid part of the cell, and it goes to the ribosome, also the rRNA. Okay, so the ribosome is made up of our rRNA. So at that ribosome, our mRNA is converted to protein by combining amino acids together based on their codon pattern to form a polypeptide. And a polypeptide is just simply a chain of amino acids. So proteins are usually made up of several different polypeptides folded to make the protein. Okay? So basically, the steps of this process is that our ribosome will grab our mRNA from out in the cytoplasm. The tRNA brings the amino acids to the ribosome. The tRNA matches with the complementary mRNA, so our codon to our anticodon. And the ribosome makes peptide bonds between the amino acids and breaks the bonds between the tRNA and the amino acid. So it frees the amino acid from the tRNA. A peptide chain continues to grow of these amino acids until the ribosome reaches a stop codon. Okay, and at our stop codon, the protein is complete and the process is over. So this is just a visual representation representation with our transcription. We have our DNA. Our DNA is opened up. We make our RNA. Our mRNA, our transcript, is able to leave. It is read by our ribosome. It is translated where it is able to make our polypeptide chain, which makes up our proteins. Okay, so what we're going to get into a little more this week is the genetic codon wheel or chart. So if you guys remember back to when we were talking about our macromolecules, our biomolecules, we were talking about proteins and amino acids, and I said that there are 20 different amino acids. So these charts right here tell us which codes, which three combinations of letters results in what amino acid. So what we're going to be doing here is basically you'll be able to read your chart and you can discover which amino acid means what. So for example, if you had some coding that said UCG, what you could do is you could go to U, you go to C, you go to G, and you'd see that, okay, this codon is coding for serine. Okay. Or if you had UAG, you could go, okay, UAG, this means it's a stop codon. So I like using the wheel letter. It works the same way on this chart over here, except you have your first code word, your second code word, and then your third here. So for example, if you had CGU, you'd go, okay, C is our first one, so we'd go down here. G is our next one. U is here where it's lining up with our U, so it would be arginine. Okay, I like the wheel. I think it works a little bit better than the chart. When you guys do your EOC at the end of the year, both of these will be provided for you. So at no point are you going to have to memorize this. So let me just repeat that for you. At no point will you ever have to memorize this wheel or chart. It'll always be provided for you, even on your EOC. Okay, so our codons, a little more on how they work. So they're read in triplets or 3 base pairs at a time. So they're read in triplets or 3 base pairs at a time. Each 3 base combination stands for one of the 20 amino acids. So we can have multiple codes for one amino acid, but there's not going to be one code for multiple amino acids. So we know that if we go back to look at it, for example, UAG is always going to be stop. UAA can be stop, but we're not going to have a whole bunch of different things that could potentially be stop. Okay, so like, for example, UAA is always going to be stop. It's never going to be tyrosine. Okay, so we can have multiple codons for one amino acid, but we can't have multiple amino acids for one codon. Okay, so some of our important ones are AUG. This is our start codon. And then with our stop codon, we have multiple ones. We have UAA, UGA, and UAG. Changing one of the base pairs in the triplet can change the base pairing. So this is where we get into our mutations, is when we see some changes. So there's different kinds of mutations that we can make. So things to know about mutations. Mutations are random changes in the nucleotide sequence of DNA. So this may occur in our somatic cells, also known as our body cells. Our body cells are not passed off onto offspring. So if we experience mutations in our body cells, that sticks with us. We're not going to pass that on to our offspring. Okay? So if we have mutations, however, occur in our gametes, which are our eggs and sperm cells, those can be passed on to our offspring, because when we have our egg eating our sperm, that is when you are producing your offspring. Okay, so with mutations, they happen regularly. The majority of mutations are neutral. However, some can be beneficial and some can be harmful. Okay, so for example, if it's beneficial, it could be a mutation that helps or creates a trait that is evolutionarily beneficial and it'll be passed on to the next offspring. However, there's also ones that can be harmful. Most of our ones that we see be harmful are what we can account for causing cancer. So harmful mutations usually lead to cancer. This can be our mutations can be caused by external factors. So chemicals and UV radiation from the sun both can cause mutations. And it can also be caused by internal factors like mistakes during replication. So that would be just basically our enzymes making a mistake. More mutations are caused by external factors over internal factors. And many of our mutations are caught when a mistake is made, especially if it's an internal factor. And with this, with it being caught by an enzyme, it gets fixed. So that's why most of our mutations are neutral, because it gets fixed. Oh no! Sorry, my baby spit up. Okay, so gene mutations. Let's talk about the different types. So this is a change in a nucleotide sequence of a gene. So it may only involve a single nucleotide and may be due to the copying errors or chemicals or viruses. So our types of gene mutations include point mutations, substitutions, insertions, deletions, and frame shifts. So we'll talk about all these. So point mutation is a change of a single nucleotide. It includes the deletion, insertion, or substitution of one nucleotide in a gene. So for example, how we could change a word with this. So for our normal one, like our normal word could be beast, okay? If we were to experience a substitution, it could turn it into a feast by changing the B out with an F. If you insert an R, you could end up with a breast instead of a beast. And if you delete an A, you get best instead of beast. So how dramatically we're changing these words is somewhat how dramatic these mutations can be within the code. Okay, so with our point mutations, an example of this would be sickle cell disease. So if you've not heard of sickle cell disease before, this is where the blood cells in a human are shaped like a sickle instead of like a healthy blood cell like here on the right. So our blood cells, they hold our oxygen and other important gases that travel around our body. When they experience this mutation that makes them into a sickle shape, it is a very bad time. Okay. So with this sickle shape, they aren't able to carry the oxygen that's necessary. That is let go. And basically, with that, the person doesn't receive the oxygen that they need. They feel weak, they feel sickly, they feel ill. So it's not a great situation. Okay. So frame shift mutations are different than point mutations. So this just happens when inserting or deleting one or more nucleotides. So this changes the reading frame, like changing a sentence. So instead of just changing the word, we're changing the entire sentence. So as you can see here, we have deleted the G and it shifted everything over. So with a frame shift, after that mutation's taken place, everything in the line is changed afterwards. So just removing this G changes this to a lysine. It changes the alanine to a histine, and we no longer get a glycine. Nothing is made here. So this causes our whole entire protein to be built incorrectly. So if your body is trying to make a specific protein or an enzyme and it's not making the right one, then our bodily functions can quickly be messed up. So frame shift mutations, like the original could be the fat cat ate the wee rat. But if we had one where we added an A, okay, we could have, yeah, I'm not even going to try to say that. The fat cat who'd heard at, yeah, not great. Okay, so these are examples of our mutations. I would also recommend looking at the Amoeba Sisters video that's going to be attached to this to check out the different types of base mutations. Have a wonderful day. I hope it's great. Bye.